going to be dealing with physiological experiments on osmosis. In this presentation, we are going to introduce ourselves to the necessary theory about osmosis needed to understand experiments under this topic. My name is Linyi Jude Fanba. I am a holder of a Bachelor of Education degree in Life and Earth Sciences from the University of Marua, Cameroon, and a Doctor of Philosophy degree in Clinical Biochemistry from the University of Chiang, Cameroon. We will look at transport across the cell membrane with much emphasis on notions that will help us understand our experiments on osmosis. For the cell to carry out life processes, it has to receive inputs of nutrients, oxygen, and signals, and send out outputs of waste products, metabolites, and secretions. The exchange of these substances between the cell and its environment occurs across the cell membrane. The cell membrane, like all biological membranes, including the mitochondrial membrane, tonoplast, etc., is selectively permeable. The selective permeability of the cell membrane is due to its lipid and hydrophobic or water-hating nature, which hinders the passage of water, the highly polar solvent in living systems, and the charged or polar soluties dissolved in it. It thus allows only the passage of small, uncharged, and nonpolar substances. Embedded in this lipid membrane are transport proteins which transport specific polar and charged substances at specific times. These membrane proteins involved with transmembrane transport include moisture channels. These are membrane proteins having a specific static shape with a pore, which allows free passage of a specific molecule. An example is the aquaporins, which allow the diffusion of water molecules across the cell membrane. Another type of membrane transport proteins are the gates or gated channels. These are membrane proteins with specific shapes like channels, but are able to open or close in the presence of a chemical, for example, a hormone, or voltage, that is, a nerve impulse. Examples are the voltage-gated sodium ion and potassium ion channels, which open in the presence of an action potential and are essential for nerve impulse transmission. A third type of membrane transporters are the carrier proteins. Carriers are membrane proteins that bind a molecule on one side of the membrane, change the form, and carry it across the membrane to the other side. An example is the glucose transporters for transport of glucose into cells. The previously described membrane transport proteins do not need metabolic energy, that is, adenosine triphosphate, to function. They permit passive diffusion of substances across the cell membrane. This other group of membrane transporters we are going to look at require energy. They are called pumps. Pumps are membrane proteins that use metabolic energy in the form of adenosine triphosphate to move substances across the membrane, often against the concentration gradient. An example is the sodium ion and potassium ion pump on membranes of neurons, which produces the resting potential, making neurons ready to transmit nerve impulses. Transport across the cell membrane can be passive, that is, without the use of metabolic energy, or active, that is, with the use of metabolic energy. Substances can cross the cell membrane by four mechanisms. Diffusion involves the movement of a substance across the membrane from its region of high concentration to the region of low concentration. There are two types of diffusion across the cell membrane. Simple diffusion, in which substances cross the cell membrane directly through the lipid bilayer. Such substances are small and nonpolar. Facilitated diffusion, in which substances cross the membrane through specific membrane proteins. Osmosis, which is a sort of diffusion of water. Water moves across the membrane from a region of low solute concentration to a region of high solute concentration. In active transport, pumps use energy to transport specific solutes across the membrane, often against their concentration gradient. There are two types of active transport. Primary active transport, which uses energy to directly transport a solute across a membrane, and secondary active transport, which uses energy to create an electrochemical gradient by accumulating a solute on one side of the membrane, which favors the diffusion of a second solute. In bulk transport, on the other hand, substances cross the membrane in membrane-bound vesicles. It is called endocytosis for substances entering the cell. This can be phagocytosis for particulate substances, for example, feeding an amoeba, and penocytosis for liquid substances, for example, the uptake of some hormones. For substances leaving the cell, it is called exocytosis, for example, in secretion by some glands. Let us now look at the focal point of our presentation: osmosis. Osmosis is essentially the diffusion of water across the cell membrane. It is the movement of water molecules from a region of high water potential to a region of low water potential across a selectively permeable membrane until equilibrium of concentration is attained between the two regions. Only 10% of the water crosses by direct diffusion across the lipid bilayer because though water is a small molecule, it is very polar. The remaining 90% crosses by facilitated diffusion through aquaporins. The water potential, denoted cell is the energy of water molecules, the ability of water to do work, which is to dissolve solutes, or the tendency of water to leave a system because of the few or no solutes to dissolve. Pure water has highest water potential of 0, while the water potential of a solution is always negative. This implies that water will always move from distilled water to a solution from which it is separated, unless a counterpressure, for example wall pressure, is developed to stop the movement. Osmosis is conditioned by two pressures: the concentration gradient, also called the osmotic potential, or solute potential, denoted CAAS, due to solutes in solution, and the wall pressure potential, denoted CAP, which is the physical pressure developed against the entry of water into the cell by the rigid cell wall, which pushes back against the expanding protoplast. The solute pressure drives the movement of water, while the pressure potential stops the movement. The combined effects of solute potential and wall pressure constitute the water potential. The pressure potential is a positive potential, while solute potential is a negative potential, such that at equilibrium, the water potential is 0. In the diagram, we see that when a cell which has solutes and thus a negative solute potential is placed in pure water, whose water potential is 0, the solute potential then drives the movement of water molecules into the cell, leading to the expansion of the protoplast. The expanding protoplast pushes against the rigid cell wall, which develops a counterpressure, the wall pressure, which equalizes the solute pressure, stopping the further entry of water. In situations involving osmosis, a tissue can end up in 3 types of osmotic environments: isotonic solution, hypotonic solution, and hypertonic solution. Let us first look at the isotonic solution. Isotonic solutions are solutions of the same solute concentration or same water potential, separated by a selectively permeable membrane. If a cell is placed in an isotonic solution, that is, a solution whose concentration is same as that of the cell remains stable because there is no net movement of water molecules between the cell and the solution. That is, the amount of water entering the cell equals the amount leaving. A solution with a high water potential has much water with respect to the solids in the solution, and thus corresponds to a solution of low concentration or hypotonic solution. If a cell is placed in a hypotonic solution, water enters the cell by osmosis, making it to swell. In an animal cell which lacks a cell wall, for example, the red blood cell, this will lead to lysis, in which the cell explodes. While in a plant cell with a rigid cell wall which can resist influx of water, this influx of water leads to a turgidity. The cell becomes fully distended and stiff and is said to be turgid. That is, it develops turgor. On the other hand, a solution with a low water potential has less water with respect to the solutes in the solution, and thus corresponds to a solution of high concentration or hypertonic solution. If a cell is placed in a hypertonic solution, water moves out so the protoplasm shrinks and collects in the center of the cell. In the animal cell, for example, the red blood cell, this leads to crenation, in which the cell membrane becomes wrinkled. For plant cells, the exit of water from the cell leads to plasmolysis, in which the cytoplasm shrinks and collects at the center of the cell, away from the cell wall, and the vacuole virtually disappears. The tissue with plasmolyzed cells becomes soft and is said to be flaccid, a condition called flaccidity. At the level of the whole plant, plasmolysis is manifested by wilting, in which the cells lose their turgor and the plant structure droops. Here we are going to look at some important points to note. Osmosis occurs only in living tissues. Since osmosis occurs mainly through aquaporins, which are proteins in the cell membrane, killing the cells by boiling a tissue or by treatment with a corrosive solution such as sodium hydroxide will denature these proteins and they can no longer perform their functions. This is why controls for experiments in osmosis entail killing the tissue, thereby eliminating the factor living. In reporting experiments during your examinations, bear in mind that though during classes solutions are labeled with their names, in examinations the solutions will be coded with letters say A, B, or C, and you will be using only the terms hypotonic solution and not distilled water, hypertonic solution and not sodium chloride solution, and corrosive solution, not sodium hydroxide solution. To identify the solutions when answering questions on osmosis, you need to assess the changes in texture and appearance for strips of tubers, the changes in curvature and appearance for strips of cocoium stalk, and changes in the stomata for leaf epidermis. We will direct you on this when treating each experiment. We have come to the end of this presentation. Our next presentation will be on the effects of osmosis on a strip cut out from a tuber. Thanks for your time. Hope you enjoyed following up the presentation. Please subscribe to this channel, like and share. By subscribing, you will be getting notifications when new presentations are available. And